Okay, so I think we are live now. So hello everyone. Thank you very much for joining us in this first talk from the Physio webinar. I'll share my screen very quickly. So this event is organized by the graduate students of the Physiology graduate program of the Institute of Biosciences of the University of São Paulo. So this is our team. Okay, you can show here. So this is our team. And we organized 4 different talks that will happen during October and November with speakers from different countries talking about themes of physiology and the intersection with other areas. So please follow us through these links and to get more information, and please do not miss any of these talks. They will be very interesting. So today we are very pleased to welcome here Dr. Enrico Rezende, who is a biologist by the University of Chile and has a PhD in ecology and evolutionary biology by the University of California. Currently, he's assistant professor at the Department of Ecology of the Pontifical Catholic University of Chile. His research is focused on the interplay between physiology, ecology, and evolution, using theoretical and experimental approaches to investigate how climate change affects the distribution of different animal lineages, as well as the composition of ecological communities. So, Enrico Rezende, thank you very much for your presence here today. After your talk, we will have 30 minutes to take questions from the audience. So everyone can ask questions through the YouTube chat, and they can also be in Portuguese if you feel more comfortable. And our team will select them and send them to me. So I think now, okay. The last detail is that this event will be recorded and available through YouTube after the transmission. Now with no more details, I'll give the word to Enrico Rezende, and we'll be here in the background in case of any technical problems.
All right, Milene, thank you so much. And thank you for the organizing committee to invite me for this. It is a real pleasure. It's actually, I think, my first or second presentation in Brazil, which is ironic considering that I am Brazilian myself. So for the students, if you eventually need to ask any question in Portuguese, I have no problems with that, all right? Actually, I'm speaking in English just because the organizing committee asked me to, all right? So let's start with the presentation. Let me see if I have here, I can share my screen. There you go.
Okay, we can see your screen.
You can see my screen, right? So it's full screen right now, the PowerPoint, right?
Yes.
Excellent. All right. So essentially today, I'm going to talk about thermoecology and the impact that, this knowledge has nowadays on our predictions regarding what's coming next with climate change. And, oops, let me see here. There you go. So it's actually a very fortunate or a privilege to be the first one talking, since Raymond Huey, he's a very big shot in thermoecology in general. He's going to be the next one talking in this line of seminars, which I just think it's wonderful. I'll probably be watching it too. And so let's just recapitulate a little bit about climate change. And I'm going to try to make it very succinct because we're a little bit limited on time. So essentially what we have, climate change, is this broad phenomenon, right, that is widely known as global warming, even though the terminology climate change may be a little bit more appropriate and more general, because it's not really for sure that everywhere on Earth will actually warm up. That's number one. And second, we'll have many different responses in terms of climate, aside from just an increase in average temperatures. And if you look at this panel, what you're seeing here is one of the plots that was available for the International Panel for Climate Change, which is this huge global community that is hired by the UN to assess the perils of climate change and to put all the information together into this vast report, right? So there you go. And what we have here is essentially the predictions of different models of what might be coming in the future, right? So this is this plot may be a little bit out of date now. This is the IPCC report from 2014, but what you see is essentially this, that from now, let's say 2020 to the future, what most models are saying is that the temperatures will increase, right? And what you have there is just the variability across different models and across different scenarios. So you might have, for instance, in orange, the internal variability, which means that the same models and same assumptions provide some amount of noise regarding what's coming in the future. Then you have the spread across different models, which means same assumptions, but different mathematical models can give you the spread, which is shown in blue. And eventually you have now the different RCP scenarios, and this means the amount of greenhouse emissions that you have in the atmosphere, and therefore the global warming might be more pronounced or less pronounced, depending on what society decides to do. And this is the source of variation that you see there drawn in green. Now, you can see a couple of things, right? So the first one is that global warming is coming, so that means that temperatures are indeed rising, and all the different scenarios, they predict that to a higher or lesser degree that's going to happen. But you do have a lot of variation, all right? So the big question then is how we can actually react to that, how we can respond to that. And at least in my personal perspective, I think the most important thing that we can do as scientists is try to understand the phenomenon and then try to predict what's coming, right? If we know what's coming, then we can try to, design strategies to mitigate these potential problems. So if you have now these changing environment, right? Let's say it's just essentially temperature, even though we have many more responses. If you have a changing environment, essentially there are three types of responses that an organism can do when faced to this kind of scenario, right? The organisms, they might adapt, so they can actually tolerate a particular environment. They might migrate, which essentially means they have to leave and go somewhere else. Or in the case they cannot migrate and they cannot adapt, essentially that lineage, that population goes extinct, all right? So from an evolutionary perspective, if you're thinking in a local problem, let's imagine what's happening here in São Paulo, actually I'm in Santiago, you might have these two alternatives, right? The animal might migrate or they might go extinct. We want neither of them. Essentially what we want to do is to understand how they can adapt to these challenges. And so the main focus that I'm going to be talking now is essentially how different organisms, populations, and then communities might be able to adapt to different, to the challenges that are coming with increasing temperatures. And so you can now decompose this adaptation into three different components, right? One is that you already have the capacity to tolerate what's coming. So that means that your tolerance exceeds what's necessary for present conditions. That's number one. In the second case, you might adapt via phenotypic plasticity, which means that essentially your phenotype changes, but there's no change whatsoever in the genotypic constitution of the population, so genes remain essentially the same. Or finally, you might evolve in response to climate change, and that would be Darwinian evolution, right? So we're talking now about changes in the gene pool and essentially evolutionary responses to this new pressure associated with climate change. Now, is that really a problem? Should we be concerned about this? And the data tells us that yes, it is a problem. You can see, for instance, that this paper by Urban in Science in 2015, it shows that the predicted amount of increase, it has a major impact on the percentage of extinction that might be coming, right? So the warmer it gets, the more lineages go extinct. And you can see this comparing the X versus the Y axis. And if you see, for instance, predicted extinctions at the top of the panel, you can see that for current temperatures, which is essentially slightly around 1.5 degrees above industrial levels, you have something on the sort of 2% of the predicted extinction. That means extinction associated primarily with climatic events. We're not talking about habitat degradation, pollution, etc. It's just climate change, right? So 2.8% of all organisms would be, threatened by extinction, given this amount of climate change, or this amount of temperature, 1.5 above pre-industrial levels. Now, if you consider what would be the RCP 8.5, which is essentially a sign that tells you that there would be business as usual, lots of CO2 in the atmosphere, and the temperature would increase dramatically until the end of the 21st century, what you find is that the predicted extinctions could go up to, let's say, 50%. which is an enormous amount of extinctions, right? Considering only this source of stress. So we do have to be concerned about this. Of course, we need mitigation strategies, but also we have to consider the possibility that many organisms might be able to adapt. And one big question is, how do we identify who's able to tolerate what's coming, and who are the, which are the species that are more vulnerable to future scenarios? And in order to do so, what most evolutionary ecologists or thermobiologists have been doing is employing this conceptual framework that you have in this slide, all right? It's called a thermal performance curve. And essentially what you have there is a representation of how performance, that means how a particular trait, it could be fitness, but it could, so reproductive output, it could be also running performance or many different proxies. I'm going to talk about this in the next slide, but it shows how these different performances change as a function of body temperature. And what you can see here is essentially that on the left side of the curve, you have an increase, right? Actually, if we're strict, it would be something near to an exponential increase because we're talking about thermodynamics here. So essentially, biochemical rates tend to increase, and therefore everything that follows thermodynamics increases as well. And so you have an increase here from CT min, which is the critical minimum, up to an optimal temperature. That's when you maximize your performance. And then after that, another process takes place, which again, in biochemical terms, would be something like protein denaturation, and essentially many proteins start losing function, and so the system starts slowly to collapse. And eventually what you have now is a decrease in performance, and then you reach your critical thermal maximum, which is shown there as CT max, all right? And so the description of this curve, it gives you many different phenomenological variables to study how temperature will affect organisms. It gives you the range of thermal tolerance, right? The minimum temperature that an organism or a population can tolerate, which is CT min, the maximum, which is CT max, the optimal temperature for that population, and of course the breadth, okay? And so this approach has been used, let's say for now, almost 50 years. All right. And it's been widely employed now in the past decade to study things such as safety margins, which is essentially how far the thermal breadth or the performance curve of a given population is from predictions or forecasts associated with climate change. If it's very far away, then the organism can tolerate, let's say, rising temperatures. So there's a lot of safety margin. If it's very near, then we're in trouble. Right? That means that a small amount in temperature could lead the population to extinction because the temperature would be above the critical limit. And so this is the general scenario, and I'd like you to, please recall this because we're going to be talking about it, and actually we're going to start criticizing this framework in the next slide. Now, why do we care? All right, so I'll do let's say, a brief overview of what we have learned with the thermal performance curve, all right? And I'm gonna show two studies which I find very, very elegant and quite nice. And so the first one, let's take a look here at the slide. Essentially what it shows is that climate change in terms of temperature, all right, is more pronounced at high latitudes. All right, you can see, for instance, in the northern hemisphere there, you have an increase of up to 2 degrees, and that's like the red region of the plot. And what it shows then is that near the two poles, the north and southern pole, what you have is an increase or in climate change or in the effects of climate change, in rising temperatures. All right, and so at the very beginning, many biologists were arguing that because the impact of climate change in terms of temperature is going to be more pronounced at high latitudes, then what you're going to see in terms of ecological systems is going to be essentially a reflection of that. So that means that systems at higher latitudes would be more threatened. Now, when we include this information, when we overlap this information of temperature with the thermal performance curve, what you see is a very different scenario. I'm going to be talking about two studies here that study different aspects of the thermal performance curve, all right? So the first one here is a paper by Dylan in Nature. And essentially what it shows is that because the thermal performance curve has this exponential increase in the left side, as you can see here in the plot, essentially what happens is that the similar amount of increase at cold temperature, let's say a 2-degree increase in a cold temperature, is going to have a smaller impact than a 2-degree increase at high temperatures, right? And you can see, oh, let me check. Can you guys see my mouse? I'm sorry,
Yes.
is there? Yeah.
Yes.
Oh, you can see my mouse. All right. So that makes my life way easier. So what I'm talking about is here, you have the same amount of increase in temperature. In this case, it's 5 degrees. And you can see that in a cold temperature, an increase in 5 degrees has a very small or negligible effect in metabolic rate. Now, the same increase, which is now in red here, you can see that it has a massive impact in energy expenditure, right? So if performance in this case, it's metabolism, essentially how much energy you're using, and therefore how much energy and food you need, essentially the same amount of increase in temperature would have a disproportionately higher effect in the tropics, because the tropics are warmer, right? And that's what you see here. Essentially, in panel A in the left, what you have is the change in temperature. And as you can see, the Arctic is warming up more than tropical environments. Now, when you transform that into metabolic costs, what you see is that the ranking changes, and now tropical communities are more threatened because of this non-linearity here of the thermal performance curve, okay? So that's one problem, and that's one, let's say, insight that you only get when you have the thermal performance curve you take into consideration. Let's see another example. Now let's go to the other side of the performance curve, above the optimal temperature. Let's go at temperatures where you're near the limit, right? Essentially what you have, and now that's another paper, that's a very important study that happened in 2008, what Deutsch and colleagues, they detected was that temperate and tropical animals, they exhibit differences in their performance curves as shown here, but the difference is as follows. In temperate organisms, that means cold adapted organisms, what you have is a broad thermal range. You can see that the curve is broad, right? Whereas in tropical organisms, the curve is narrow. Now, when you compare CT max, you can see, and I've shown this in the slide here, that the CT max are very similar, all right? They do not tend to differ that much. And so what happens is, when you include again those climate forecasts, you overlap that with this sort of information, what you get now is once again an inversion of what would be the prediction, right? You'd expect that climate change is stronger at high latitudes, but its impact, it's going to be disproportionately stronger in tropical environments, all right? And that's what you see right here. Essentially, you can see that the impact now in these two graphs, essentially they show the same thing, but is negative at lower, let's say negative, it means it's detrimental, it's dangerous, all right? At latitudes near the equator here, the 0, all right? So that means that high latitudes are not so threatened by climate change, even though the weather is going to change quite a lot. Whereas tropical environments where climate change is not going to be as pronounced, it's going to have a massive impact in ecological communities, which is a problem because it's actually the regions where it's more diverse, right? And so I hope to have convinced you with these two studies that which both of them show the same general pattern, which is that the direction that you'd predict that the geographic pattern that you'd expect from climatological studies, which is a more vulnerable environment at high latitudes, it's actually inverted when you include information on thermal biology, essentially this information on the thermal performance curve. Now, here, these are, let's say, I would put it this way, these are now classic studies, all right? Now, there's a problem with the thermal performance curve, and I'll list 4 major limitations, all right? It's a phenomenological approach, which means it describes the phenomenon, but it doesn't really take into consideration the mechanisms that give rise to this phenomenon. It employs static endpoints, which means the critical thermal limits, CT min and CT max, are a value and they're constant, right? That means a given population tolerates, let's say, 30 degrees, not more, certainly less, okay? And so that means that the endpoint, it's static, it doesn't change, it doesn't vary. And for these reasons, it has a low resolution, which that's the reason why I have the European continent here in the top panel in low res, all right? And it has a low predictive power. And you can actually see that in the examples that I told you, all right? Because the phenomena that I showed, or let's say the 2 large results, they happen on a global scale. So that means we're comparing, let's say, organisms and communities between the poles and the equator, which is fine. You can get these macro scale effects. But here's a question. What might happen to organisms, let's say, around São Paulo, in São Paulo state, if suddenly we have something like a heat wave? all right? It's a very local phenomenon. It happens in 2 or 3 or 4 days. And then we cannot employ thermal performance curves to work or to come up with some predictions, because the predictive power and the resolution is just very low. All right. I'll put something else here. Another problem that is very frequent in the literature is that ideally, if you want to talk about vulnerability of populations to climate change, essentially you have to deal with fitness, right? Essentially what you want to know is whether the population will be capable of sustaining itself in in evolutionary time, or at least ecological time, all right? But that's very difficult to measure. And therefore many researchers, they have employed proxies. That means they have measured other things, such as knockdown temperatures or loss of motor coordination, to infer what would be the critical thermal limits of fitness, all right? And that's a problem because the proxies might not be very good. So that's another level of limitation, right? That might be something else, a confounding effect that might be also decreasing the resolution of our predictions. And let me give you an example. Coming back to the original finding by Deutsch, remember that I mentioned that CT max in tropical and temperate species is roughly the same, right? And actually here, they did, Miguel Araújo and many colleagues, they did a meta-analysis, and they show here, which is in orange, that CT max, it exhibits very limited variation, all right? Which is a similar result to what Deutsch had. And other papers until now, so maybe till the last, let's say 4 or 5 years, they would claim that CT max did not differ across different geographic locations. And so what you have now is this idea that CT max is highly conserved across lineages and across the whole planet. Okay, and therefore, like Deutsch, you would see similar CT max for temperate species and tropical species. Now, that would imply, and many papers have been talking about this for the past 10 to 15 years, that CT max has, it cannot evolve quite a lot, right? And so essentially, some people have postulated that we're talking about a physiological limit. Studies on the heritability of this trait have shown that it has very low heritability, which means that even if it's under selection, it cannot evolve, it cannot respond to selection. It varies very little between populations and species, and there's no geographic trend whatsoever, right? So that means, according to these studies, essentially what we have is a single CT max, or that varies very little across organisms, which is concerning because if temperatures are rising, essentially all organisms will be equally threatened, if you will. Now, we have argued that this result actually reflects the low resolution, a problem with statistical power, and not actually a problem with, let's say, the biology. We argue that many organisms could respond, and let's try to put it to a test. So the big problem that I've been working on in the past 10 years is the fact that the endpoints, as we call them, right, CT max and CT min, they're not static, right? They change. And why is that? The reason is, let's imagine if we're in a sauna and you ask, so how many degrees can you tolerate in a sauna bath? You can say, well, look, I'll tolerate 45 degrees, but if you put me in a sauna bath at 50, I'll probably die. And what is missing in that example is for how long. I might be able to tolerate 45 degrees for 10 minutes. I might be able to tolerate 50 degrees for 1 minute, if you will. So the thermal stress, it's actually a cumulative effect. And therefore you have an interaction of two components. Of course, there is temperature, but there's also time. And what has been missing from previous studies, or so we argue, is that a static endpoint is not a good description of thermal tolerance. You need at least 2 dimensions to describe thermal tolerance. You need temperature, which is the intensity of a stress, and you need time, which is the duration of the stress. And you can see here in these graphs, and that's empirical data from Maynard Smith, that there is a predictable equation that shows how these 2 components are associated. Essentially, what you have is that the amount of time that you can tolerate a stress decreases in a log linear way with intensity. All right, and you can see that there. So that's 2 ways you're plotting the graph here. You have temperature increasing, right? So it's becoming warmer and warmer. And you have the amount of time that you can tolerate that temperature decreasing, right? And the panel on the right is exactly the same plot, but now shown in absolute scale. So knowing log scale, you have log scale in the left, absolute scale in the right. And the other thing that you see here now is that instead of having a single point that separates life and death, you have a function, right? That separates the region where you can tolerate stress from the region where you can, or you eventually die, right? Now, it's a little bit more complex than that, because as I mentioned here in the caption, we're talking about survival probabilities. And so essentially, the line that I showed you before assumes that everybody either lives or dies, right? You're alive below the line, you're dead above the line. But there's a problem with survival probability function. And the reason why I changed the X and the Y axis, right? Normally we say the independent is the X axis, the dependent is the Y one. The reason why I interchanged them is because the real dependent variable is the probability of survival. And the probability to survive depends on both time and temperature. And actually, the graph that you'd be seeing is something in 3D, all right? So the lines that I showed you in the previous slide actually correspond to this LT50 right here, all right? We are assuming a given probability. And because we're using average values, we're assuming, let's say, the lethal temperature for the 50% of mortality. So how do you interpret this 3D model here? Let me see if I can put just a moment. Oh, no. Oh, there you go. Let me see if I can annotate here to make everybody's life easier. Oh, annotate, there you go, all right. So how does this work? Let's go with the first curve here in the left panel. What you have is, let's imagine you get 100 individuals of a given species and you put them at 30 degrees and you follow how they start dying. Essentially, everybody's alive, so that's 100% survival. And then slowly they start collapsing and eventually they get to zero. So that's the curve highlighted in red. So that's the survival probability curve for 30 degrees. That's a constant temperature. And then you can do the same thing for 31 degrees, right? And there you go, 31 degrees now in red. And then 32 degrees and so forth. So you arrive at 35. And you can see now that this curve that you have on the left side is actually the same 3D model that you have here. You're just slicing the surface in this way here, all right? And so essentially, it's just different ways of representing the same phenomenon. And the phenomenon is, how does the probability of survival changes as the temperature and the time combination change accordingly, all right? So now that we have this new framework, let's see if it works. Let's see how it performs with some data. There you go. So what I did is as follows. I downloaded all the data from two major reviews, which was Hoffman and Araújo, which I showed in the previous slide. And I tried to look for latitudinal clines as they did. Essentially what I did is, first, I ran a single model, which is CT max as a function of latitude right here, all right? And what you see is that latitude has no effect. So I would get to reach the same conclusion as they did, right? So essentially that there's no latitudinal cline, but you can see that the amount of variation that I can explain from CT max is tiny, an R-square of just 0.21. So that means 2.1% of the variation is explained with this model. Now let's see if my model works. So we include now the logarithm of time in the model, right? So there you go, the second line in the table. And what you have now is that time is highly significant. And the R-squared, the amount of variance that I can explain now with this model, increases to almost 64%. And now let's add latitude, because now we're controlling for the effects of time. Now let's include latitude in this model. And that's the third line. And there are two interesting outcomes here. First, of course, now the explained variances increases. So now you have 68% of the variance is explained with the R-square, but also look that the probability of latitude, or the effect of latitude now, is statistically significant, all right? So there are two take-home messages here. First, you include time, or you control for time, and you're explaining a lot of the variation in published values of a CT max. All right, that's number one. So that's something that people have been missing for decades. And the second component is that when you do the final analysis, so the last model, and you check the slope of this regression for latitude, essentially what you get is this, that heat tolerance increases around 0.15 degrees Celsius per degree of latitude. All right? Which means that if you compare an animal from the equator, latitude 0, versus an animal from high latitudes, let's say latitude 60, their difference in thermal tolerance, in heat tolerance in particular, is almost 10 degrees. And this is for constant temperatures, all right? So it's not a small difference. It's actually a very high difference. And now what you have here is that there is adaptive variation for heat tolerance, and we've been missing it for quite a long time. And the reason for that is that low resolution. We've been using an incomplete approach, if you will. Now, as I mentioned, all these measurements, they involve constant temperatures. So the tolerance landscape that I showed you before, in order to validate it, and when we include log T in the analysis, it's always assuming that temperature is constant. Now, that was a major limitation, right? Because in natural populations, we have varying temperature. And so we need to be able to develop a predictive tool that deals with temperatures as they are in nature. And this is what we've just done. So this is a recent paper that came out in Science now, maybe a month, 2 months ago. And what you see here in the left two panels, so we're talking about panels A and B, is the same description that I showed you before. It's exactly the same plots. You can see it's constant temperatures, and we have here an animal measured in, or population if you want, measured at 3 different temperatures, temperature 1, 2, and 3, all right? And then you have that association between the log time and the temperature, just as I explained before, and you can express that just as these curves that fall, as I showed you in the previous slide. That's all fine and good. But now what happens when the temperature increases? And what we have shown is that you can employ calculus in order to come from these curves here in panel B, which you see there blue, purple, and red. You can use the derivatives to check how the slope would change as temperature slowly increases from T1 to T3, as shown in here. I hope you can see my mouse. So now the temperature is not constant, it's increasing from T1 to T3. And as the temperature increases, the slope of this curve slowly changes from the curve here in blue to purple to eventually red, all right? That's the theoretical framework. It's essentially just pure calculus. That's how we let's say, we expanded now the model from constant temperatures to variable temperatures. The big question is, does the model work? And so what we did now is we obtain measurements from animals measured at constant temperatures, all right? So in the lab, just as the typical measurements that I showed you before. And we employed these measurements to predict when and at which temperatures these animals would die at variable regimes, when you put them, for instance, in a bath and the temperatures keep increasing. And so we did this for 11 different species of flies and different warming temperatures. You can see here the warming rates, 0.25 degrees per minute, 0.10, and 0.05. So that means the one here at the bottom is very slow, is very fast, so animals die fast. And the one here at the top is very slow, animals take a long time to die. And what you see in the x-axis is the amount of time we predict, according to our model, that it would take to kill them. And there you go. In the y-axis, we have the measurement, right? And the prediction works incredibly well, all right? So our model, based on pure calculus, predicts essentially when the animals are going to die extremely well. And when we do the same thing for the temperature, all right, so not only when they die, but at which temperature they should die, note that the model also predicts extremely well what's coming in ramping experiments. And there you go. In both cases, you have an R-square above 0.95. So the models work, let's say, remarkably well. And the question is, if we can do this for laboratory experiments, can we predict what would actually happen in nature? We try to do so. And in order to put this to a test, here's what we've done. We collected temperature data from winter to summer, in this case, and this is central Santiago. So these are the temperatures from Santiago, Chile, all right, on an hourly resolution. But in order for our model to work, we had to interpolate temperatures on a minute basis. We need now a temperature resolution working on a minute scale, okay? So we have now the temperatures on a minute with a minute resolution. And essentially what we try to do is to predict when these animals or when organisms are supposed to die at different days. Okay. Now, just to put in comparison here, what we have here shown as critical temperature in warming assays, that's what most people would obtain as CT max in previous lab experiments, neglecting all the framework that I've mentioned, all right? So in theory, because of CT max as we see it here, nobody should die. Essentially, that's that, right? The temperatures never reach CT max, then you should have essentially no organism should die given CT max and these temperatures. Now, let's put our model to work. So what we did is we measured thermal tolerance, or the landscape, in this species of Drosophila. We measured it in cold acclimation and in warm acclimation. So there you go. It's cold acclimation, it's 13 degrees in blue, and warm acclimation, it's 18. So that's just phenotypic plasticity. There's no, it's the same population. We just want to see how acclimation works, or how it affects thermal tolerance in the field. And you have the predicted daily mortality here for cold and warm acclimated individuals. All right. So you can see, for instance, that cold acclimated individuals, they start dying, following that thermal stress from the previous slide in October, and eventually by December, you see that daily mortalities are pretty substantial. They can reach up to 60% of the population is dying in a single day. All right. In contrast, warm acclimation allows for a better tolerance, if you will, and then you just have a substantial mortality starting in December, which, well, most of you are Brazilian, that's the beginning of summer, right? So that means, first, warm acclimation definitely has, let's say, an effect. It's definitely important. Now, what happens when we do the cumulative effect? Because these are mortalities on a daily basis, right? Now, what happens with the population day after day? We can simply multiply them, right? You multiply the individuals that survive day 1 by the individuals that survive day 2 and day 3 and so forth. So you can actually plot the cumulative survival predicted by the model. And this is what we have here, all right? Now, let me try to explain this panel a little bit. First, in blue and red, we have the warm and cold acclimation that I showed you before, all right? That's number 1. Second, we use now temperature records for 2 different periods, from 1984 to 1991, that means 30 years ago, and 2014 to 2018, that means now, all right? And what you see is that the predictions of cumulative survival, or mortality, if you will, right? Because here you have 100% survival, and then as time goes by, the population starts dying because of temperature, according to the model, all right? The dotted line shows what would be predicted in 1984 to 91. But then 30 years later, due to climate change, due to difference in the thermal environment between these 2 periods, you see that the window that these organisms can tolerate decreases. They're dying sooner, if you will, right? Both warm and cold acclimated. And that's shown in the arrows. The red and the blue arrows, what they show is the impact of global warming in this population of Drosophila here in Santiago, all right? Now, that's all fine and well, but the question is, does it mimic what you see in natural populations? And to put that to a test, what we did is we collected information from another researcher that he did a longitudinal study that lasted 8 years, just checking the abundances of different Drosophilas here in central Santiago, all right? And he actually did that between 1984 and 1991. So that, and what you see is in the shaded area, which is field abundance. We standardize the abundance on a yearly basis, so the maximum is 100%. So the top abundance of the month, let's say, we set it to 100%. And what you see is the following. This Drosophila, which is Drosophila subobscura, it starts increasing its abundance at the end of winter. It increases exponentially, as you'd expect, right? It's a population increase. It's just the intrinsic rate of increase of a population. So it increases its numbers exponentially every year. And then around October, November, when our model predicts that you should have thermal death, you should have mortality due to temperature, you can see that the population stops increasing and it starts collapsing. And the rate of collapse of the population is in accordance to what we predict. All right. So this is how we put our predictions now to a test with a field population. And as you can see, our model does a very good job predicting when these, this species in particular should die throughout the year. And in contrast, remember that in the first slide I showed that CT max, you'd expect that nobody would die. And what you show here is that clearly many organisms, or the whole population, is collapsing, apparently because of thermal pressures. Okay, so now we have developed a good model, and we validated the model to study thermal selection in the field, in natural populations, employing high resolution temperature data. What else? Another problem that we have in the literature regarding the use of CTmax or endpoints, thermal tolerance endpoints, is that, as I said, not only these studies, they did not detect latitudinal effects, they also didn't detect any effects of scale, any effects, allometric effects, right? So body size effects. And so we tried to revisit that with a colleague of mine in London, and we went back again to the literature, and we asked the question, does thermal tolerance, or heat tolerance in this case, vary predictably with body size? And we tested this question in 2 different data sets. So you have the endpoints here shown in A, that's one data set, and you have here the thermal death time curves. So the association between mortality at temperature and time, which I showed, let's say, the framework that we developed with some colleagues. And these 2 data sets here, we analyzed them in parallel. All right, and you have the outcome here of the 2 analyses in the right panels. And essentially what you can see, let me try to, you have now the scaling effect here, but the main take-home message here, I'm not going to expand too much on this one, is as follows. The general curve, let's assume a constant probability survival here, so 50% for instance, the general curve of thermal tolerance, it does scale with mass. Now, it's not the typical scaling that we know, right? It's not an allometric equation as usual. What you have now is that the function, the association between temperature and time will vary depending on body size. And what happens, let me try to describe the panel, is that small animals, small organisms, they can tolerate extreme temperatures, sometimes even higher temperatures, for a shorter amount of time. And that's why, for example, when we see extremophils, right, so some bacteria that can tolerate very high temperature, it's not surprising that they're small. Because the problem is that they can tolerate that for a very small amount of time. And then if that stress is prolonged for a long time, its tolerance decreases very rapidly. In contrast, large organisms, what they do is they cannot tolerate those acute changes. So their thermal tolerance to an acute stress is more limited than for a small one. Now, they can tolerate that for longer periods of time, all right? I'd like to use the analogy here of a sprint runner. You can tolerate an acute thermal stress for, that's very rapid, but your stamina decreases very rapidly as well. So you're a sprint runner, right? That's a small organism. Or you're a marathon runner. You cannot run that fast, so you cannot tolerate something very acutely, but you can run for a long time. And so you can see here how the curves, they change, and now they change predictably according to body size. So now, not only we have seen latitudinal effects in thermal tolerance, not only we have improved the capability to predict how thermal tolerance will impact populations in the nature, now we've also been able to unravel scaling effects that have been also not detected, to say, all right? So, coming back to the main criticism that I said, that thermal performance curves, they were phenomenological, static endpoints, et cetera, let's talk a little bit about what's coming in the future. So given these limitations here, what I can see now looking back in history, what many, great scientists, many good people have done is normally they have employed ecological information, right? So geographic distribution, etc. And they have used ecology to try to explain physiological variation. So for example, they come eventually to the conclusion that organisms at high latitudes, they may exhibit a higher or a lower metabolic rate, for example. So that's ecological information being employed to predict physiology. What we're trying to do now, what I think with climate change, there's an urgency to start improving this framework and change a little bit. So we need more mechanistic models, right? They cannot be static endpoints anymore. They need to be dynamic, because dynamic models are more realistic, number one. They have to be probabilistic, right? Because we need to include things like the probability for a population to survive. Remember that selection is probabilistic, right? It's the probability that a fraction of the population will tolerate the selection event, okay? And these models need a higher resolution, and they have to have, of course, a higher predictive power, which I showed you at least one example. So with this new framework, we can now close the cycle, and we can start employing physiological data, physiological information, such as thermal tolerance measured in the lab, to predict ecological patterns, such as when is this population going to be under thermal stress and which populations might be vulnerable to climate change, okay? And in this sense, we have now the tools to increase the temporal resolution dramatically. So I'm showing here, we have now, we can incorporate things like temporal and spatial variation in temperature, time in the left. You have these highly, very high resolutions in temperature profiles. Now, as I said, we can come up to the resolution of minutes. You have that this picture in the right is 1 kilometer square. So you can see how the microhabitats will vary in that very, very small amount of area. Okay, so we can improve now the predictions, including temporal variation, and we can improve the predictions, including spatial variation, and come up with better mechanistic niche models. We can also try to put together things that have been generally studied separately, such as patterns of geographic distribution and the coexistence of species in a single community. For example, in this case, and we're working on this right now, we have different Drosophilas that are distributed across the latitudinal range in Chile. Now, of course, with climate change, as temperatures increases, most of these populations are going to be pushed south, right? They're going to leave, let's say, northern populations, especially because the north of Chile, it's a desert. And so many of them are going to be driven to the south. And that means that they will start overlapping more and more. And the question is, if we have information on distribution, can we understand how they'll start interacting between themselves in a community? And so here, one of the ways that might be mediated is by thermal tolerance once again. So for these species, we have measured thermal tolerance here, the panel B in the right. And we have also tracked their periods of activity throughout the different years. And so you can see, for example, here that you have 2 Drosophilas and they can tolerate different thermal windows. Now the CT mean and CT max, it's actually the one inferred from field population data. It's not measured in the lab. It's the one that we can infer when populations increase in the field based on environmental temperatures in the field. Oops, sorry. And so the idea is to eventually be able to put these 2 components together and try to come up with ideas to predict how these populations that are essentially moving away from the equator, they might overlap in a community and they might, coexist, right? How they can eventually build up new communities with new overlapping species. And we can get into further detail by including also information on species interactions in this sort of data, right? So not only the species are in the same communities, but we can also try to include things like the nature of the interaction between these species. And then we can come up, for example, in this case, I used performance curves as just a good proxy for the scheme, but you have something like the climate forecast for the future. And then you see that the temperatures, they might, in some cases, they might predict that both species will coexist in a given place, but not in the other. See that in this case here, in 2050, only the red species survives, not the blue ones. And when we overlap that to, in this case, a food network, any kind of interaction network, we can start understanding how thermal effects might cascade across the whole community. And then we can understand, again, have a better understanding of community ecology, employing information from thermal biology. And to summarize, I really like the statement. I think it has some like prophetic value, if you want. All right, Michael Anguilera, when he talked about thermal adaptation, the first book on the subject, his main conclusion was this, that many thermal biologists have described patterns to the point that we have gathered a lot of empirical data, but our theoretical understanding remains very, very limited. So I think now it's time that we should start trying to balance this, these 2 phenomena, right? Empirical observations and the development of theory to come up with better predictive tools for the future. And with this, I'd like to thank you all for your time. I just left here an homage to Kino, Mafalda's father, and I'll thank all the institutions that in one way or the other have been promoting my science and research. And thank you also my colleagues for all the help during these years. Thank you very much. I'm glad to take questions now. Yeah.
Thank you very much, Enrico. It was a very, very didactic, very clear presentation. I think we learned a lot from it. Yes, we have some questions here, and I read them now. Danilo Giacometti
One problem is that the perception or how different organisms will respond to that microenvironment depends on their size, right? So for example, you might have flies that can escape high temperatures, but maybe lizards cannot just because they're bigger. Okay, yeah. So there's a component which is to combine not only measurements of thermal tolerance and measurements of microenvironment, but we also need information about behavioral thermoregulation, body size, etc. But I think the main point that you can take from this presentation is that by now I think this is possible. So we can get there. All right. The main point is, in order to get there, what we need to do is to start putting more emphasis on the development of theory. I think there's just too many people in the world measuring data, measuring, let's say, CT max, if you want, or any sort of, thermal tolerance, if you want, empirical measurements, and not enough people developing good theory. Okay, so that's why I say that we have to put the emphasis, start balancing the emphasis.
Do we have now enough resolution in temperature data around the world to understand the microclimate of these animals so different?
To some extent, yes. So, for example, let me see here. I'll share the screen again if I can, because there you go. There's a button. Let me share you again the screen. All right, to answer this question. There you go. So the panel in the right. So Michael Kearney, he's showing here the microenvironments, right? The interesting thing is that this microenvironment, it's simulated. We're employing biophysical models to simulate how this environment changes depending on the amount of cover, the amount of shade, sun radiation, wind speed, and so forth. And the tools that Michael Kearney developed in particular with Warren Porter, who's an American professor. So Michael Kearney is Australian, Warren is American. These tools now, they're good enough that you can actually pinpoint different places in the planet. And if you don't have reliable information, you can try to simulate this sort of environment. All right, because the papers that they have shown, they have already validated the models. Just to give you an example, you see the panels in the left, the two panels here with the temperature. You have a black and a red profile, right? You have the data shows black and red data in these two temperature profiles, right?
Okay.
All right, so the red is empirical, the black is simulated.
Very awesome.
All right, so that means that this and one you can see here in the y-axis, one is simulated at 3 centimeters above ground. So that right, can you see me? I think it froze.
Yeah.
All right. So one is 3 centimeters above ground, and the other is 15 centimeters above ground. Same place.
Mm-hmm. Very good predictability, right?
Yeah. So the possibility to simulate different environments already there. We can do this. To simulate microclimates, we can do this as well. All right. And there's plenty of data on CT max and thermal tolerance to start combining these two approaches. Okay.
Very nice.
So yes, we can take thermal heterogeneity into account, hopefully including body size in one way or the other. But it's not only possible, it's necessary.
Very good. Beatriz Fernandes asked if these models in the expanded framework, I think in the beginning, are for which animals? And could you tell us more, how do you do to design them? I was very curious to understand the process to get there.
So answering the first question, which animals should it apply? This model, it's basic calculus. Right. So what it means is that as long as the assumptions of the model apply, in theory, the model should apply as well, because it's just like transforming velocity into acceleration. It's just calculus. All right. And so the assumption in our case is that body temperature equals ambient temperature. So that means that this model should be applicable to most, if not all, small ectotherms. Right. And now we can relax this assumption, because we could also, for example, use biophysical models to translate ambient temperatures to body temperatures and include thermal inertia, and then measure what happens with survival. All right. And we're working on this right now. So to try to explain, in principle, this model is applicable to every small endotherm you can think. And if it's a little bit bigger and it has thermal inertia, then you could include a biophysical model to translate ambient temperature into body temperatures, and then you do survival. So in principle, it's applicable to, I would say, many, many animals, no doubt about it. It still needs to be validated in other organisms, but in principle, it should be, all right? So that's the answering the first question. Now, answering your question, okay, I'll answer yours. You're asking exactly how we get to this model. And so I think that the best way to understand the model is this. You see here the 3D panel in the right?
Okay.
So you can see that this 3D panel, it actually reflects the plot on the left, which is a constant temperature. And as you just maintain animals there, they start dying and you get the survival probability. So far, so good?
Yes.
All right. So what happens when we use differential calculus? What happens now is that you can imagine this 3D wave moving. And the survival curve is a surfer surfing the wave.
Okay.
You know what I mean?
So you get differential curves from every slice of the 3D graph, right? Right.
You have the way you have this 3D surface, but if the temperature is constant, then the surface remains constant. If the temperature varies in time, then the surface is going to move. And the way it moves, the final, let's say, probability curve is going to look like a surfer falling down the wave. Okay. All right. It starts at the top, right, at 100% survival, and then it surfs its way down until 0% survival. Now the wave is moving and the surfer is falling down. And that's this panel C here at your right. This is a moving wave. And that's why it changes color.
Okay, because it changes the temperature.
Yes, the temperature is changing in time.
Okay.
And so now you have to take into account this, right? Because I said it's temperature and the intensity and the duration of the stress, but now the intensity changes with the duration.
Okay, I think I get it now.
All right, I hope you do, because I always thought the surfer example was a good one.
Yes, no, it is very illustrative, very nice. So the next question is from Carlos Eduardo Tolucci. And he asks, I'd like to ask if there are studies connecting the metabolic rate change due to temperature change with the availability of resources in those impacted environments presented here.
Oh, just a second. I've received a request here. I'm sorry. Let's say if the impact in metabolic rate is correlated with
the availability of resources in those impacted environments.
That's a good question, and the answer is no. There's no information on that. Right. Oh. So essentially, most of this sort of analysis is generally taken from the side of the thermal impact on metabolism, right? And then you estimate that if metabolic rate increases, then the necessity for energy, essentially your food requirements, increase as well. Now, the problem with trying to incorporate the amount of resources available in a particular environment is that it will depend on what's the source of food, right? Are we talking about a herbivore? Are we talking about a carnivore? And then you get into a lot of spatial problems as well, right? Fine grain, for example, a herbivore could eat a lot of resources if it's around, a carnivore might have to forage around, et cetera, et cetera. So no, the shortest answer is it's never been done.
Okay. Carlos Navas also asks, the acclimation of physiological variables to temperature is known since 1850. Why do you think it took so long to acknowledge the relevance of plasticity in this context?
I think because the big explanation is this. There you go. I think we've been stuck for a long time in the school of thought here. All right. We've been always focusing on the problem of how ecology or environmental variables, like, for example, acclimation temperature, shapes physiological responses. And so we know that animals acclimate. We know that they change phenotypically in response to different temperatures, for example. But because we didn't have any tools to go back and close the cycle, so we say, yes, they acclimate. So what? What happens? Then we need to know how these responses translate back to field conditions. And we didn't have the tools to do that, number one. And in most cases, we didn't even think that was a relevant question. All right? I think an interesting thing, and that's why I was showing Anguilera's statement at the very end, because he's showing, look, there's a huge gap in knowledge here. There's a lot of people working this problem, but they're working only in one side of the coin, right? In one side of the equation, if you want. We need the other one, development of theory, better theory, all right? And just for the sake of seeing some, the reason why I mentioned this is because this reflects a little bit of what happened during my career. So I was a physiological ecologist doing this sort of experiment throughout my career and during my PhD. And then I switched from a PhD in evolutionary physiology to community ecology and theoretical work. I worked with community ecology and interaction networks for a long time. And I was amazed that these people, they could do very, very complicated theory. And I always wondered why we didn't do that, why people in physiological ecology never went through the trouble of developing predictive theory. And so, well, there's an open niche, let's go for that, right? But then you realize that this second component here is missing. Any more questions?
Yes. Oh, sorry. I thought there was a problem with my audio.
All right.
Okay. So Patricia Takinardi asks if there is some she said a fact. There is some evidence that animals can shift their phase of activity in response to environmental changes, for example, to become nocturnal. Is it possible to incorporate that into your model? Since the time of activity can change the temperatures to which the animal is exposed, could it be considered a strategy to deal with climate changes?
Yes, that's a very, very good question. All right. So many of the previous models, because they're static, as I mentioned, right? So you have one thermal limit, for example, it's 35, period. Right? So they would use just the maximum temperatures, and normally they would use the maximum temperatures on a yearly basis, right? So they would measure or use that estimate for a given year. What is the maximum temperature in São Paulo every year? Well, it's 35, 37, let's say. But of course, now that you have dynamic models where you include time, it's definitely a possibility that you can avoid that, right? You can go and start reproducing in spring. Of course, the question remains, how can you tolerate summer? And so you may have some insects that go into diapause. It would be a possibility. You can have insects that would shift their daily activity patterns. So they might go nocturnal. Sorry, I talk about insects, but it could be any organism. All right. We have done some models. I think we published just this year an AMNET for nocturnality in mammals. And we've shown, in this case, static models, that nocturnal mammals are less threatened than diurnal ones, which you'd expect. Right? So yes, patterns of activity can change. And again, that's a general area that few people have studied. And we need more people studying it on a theoretical perspective, yeah.
Very interesting. I also thought about how this interacts with the question before, right? How temperature changes with time, and how this could be important for animals. Well, the next question is from Andresa Caeiro. She asks, those modeling were made for terrestrial species. Are there some evidence for marine species?
The answer is, to some extent, yes. So the first paper that I published on this subject, let me see if I can, the first paper, this functional ecology here, all right? The big question was, did all animals that you have in the literature comply to this relationship between temperature and time? Was it log or semi-log, right? So time, log, temperature in a normal regular scale, okay? And I found data in insects, in fish, and in many bivalves, all right? So mollusks. And the landscape is this. So essentially, yes, so many marine animals, they do show this sort of a response. And I'm collaborating with people in England, and they're doing this for organisms, intertidal organisms, right? So many organisms in the beach. And we're seeing the same result. So many snails and many organisms that live in the coast, they're marine and they forage underwater. They remain inactive at low tides, and they show exactly the same response. So in principle, as far as I can tell, across all the data that I've surveyed, these responses are general. Okay.
Very good. Also in the same direction, there's another question asking, would ectotherms and endotherms would be equally affected by these changes in temperature?
That's a good question. I'll put it this way. Ultimately, yes. Ultimately, there should be no major difference in these groups. The problem now is that the assumptions of the model is that body temperature and ambient temperature are the same for the model, right? And so, as I mentioned, if you get a larger ectotherm, then you need to put thermal inertia because the assumption of equal ambient and body temperature is not true. So you need to translate ambient temperature to body temperature, all right? With endotherms, you need the same translator. You need something that translates ambient temperature into body temperature. And normally that's the metabolic curve, right? So that means for endotherms, you would need to include something in between, which would be thermal regulation and how effective it actually is. And when thermal regulation essentially, or when temperature is no longer regulated, in which range. But again, in principle, I think the general idea holds, right? You have a survival probability curve, you have different temperatures, so eventually you have selection, thermal selection, animals dying because of heat or cold. Right? Now, the problem with endotherms in particular is that ambient temperature is not equal to body temperature. You need something in between to translate one into the other.
I imagine the proxies may be different also, the CT max, CT minimum.
Oh, yeah. People use the endothermic curve, the metabolic curve, all right? So I've worked on this as well. And essentially, you don't measure CT max in endotherms, primarily because it's not ethical, right? You cannot freeze, let's say, a bird or a rodent to death. And so, or, CT max or CT min, you cannot cook them. And so a lot of the work that has been done with endotherms has relied on indirect proxies. And in my opinion, nobody has actually validated these proxies. So we do have some broad patterns, but I think nobody actually went and checked whether the proxies are good or not to infer thermal tolerance for endotherms. As far as I can tell, that has not been adequately done.
Okay, I imagine so. Okay. The last two questions. Rafael Bovo asks, nice and clear talk, thanks. What was not so clear? How to scale up effects or impacts of, let's say, raising temperature on organisms to population community levels? This is critical since it's not so clear that, for instance, temperature effects on organisms scale up to population community level.
Yeah, so the interesting thing about this study here, right, this one, is that this is actually a population, right? So that means we measure critical temperatures at the organismal level, we see how each individual dies, but then we transform that into a probability survival curve, and that probability survival curve is at the population level, right? So in this case, we're already dealing with population here. Now, how do we translate that into a community? That's a very good question. In principle, you have to measure every population in the community, right? That's the principle. Now, there are ways to circumvent this problem, right? You could measure a few species in the community, and you can use things like phylogenetic methods to try to infer what might happen with closely related species, for example. Another way is to use scaling, right? If you measure a few species and then you know that you have allometric effects, you can start predicting, even if you don't measure thermal tolerance for everybody, you can start at least having some good idea, more or less, where thermal tolerance lies. Right? And so eventually you can come up with a scenario where you can start simulating what might happen at the whole community level, even if you don't measure everybody, which I understand it's just impossible. But there are ways to circumvent this problem as well.
Okay. So with this question, we are done now with this. Okay. So thank you very much again for your participation here. Thank you for accepting our invitation. That was very nice. We learned a lot from this. And I also like to thank everyone who stayed until the end. We hope you enjoyed this transmission as much as we did. And we hope to see you all again in our next talk. And well, that was a pleasure. Thank you very much.
Thank you. Thank you. Congratulations for the organizing committee. It was a major pleasure. And I hope you all are well and safe. All right. And please keep safe.
For sure. For you too. Ok. Acho que agora terminamos, né?